Hello students, I'm Dr. Arpita De. A very warm welcome to our lecture. The topic for today's discussion is ultrastructure of chloroplast. Before we start, let's first talk about what is a chloroplast. A chloroplast is an indispensable organelle in the cell known to play primary role in photosynthesis. It is a semi-autonomous double membrane bound structure which is known to play vital role in photosynthetic activity in green plants and green algae. Simper in 1885 categorized plastid into three classes or three types: chloroplast, leucoplast and chromoplast. Chloroplast is popularly known as the green plastid, leucoplast is the colorless plastid, and chromoplast is your colored plastids. Chloroplast is normally found in green algae and green plants, and the chief pigments are chlorophyll A and chlorophyll B. Leucoplasts are plastids that are devoid of colors and are involved in storage functions. A myeloplast stores carbohydrate, elioplast stores lipids, and proteinoplast stores proteins. Chromoplasts are colored plastids which impart various colors to different types of plants. The two most important types of chromoplasts are pheoplast and rhodoplast. Chloroplast, leucoplast and chromoplasts are interconvertible. It means what? Chloroplast can get converted to either of the two forms, that is leucoplast and chromoplast. Chromoplast can also get converted to either chloroplast or leucoplast. This means what? All these three subtypes of plastids are interconvertible. Now let's talk about the origin of chloroplast. Chloroplast is believed to have evolved or developed from proplastid. Proplastid is a double membrane bound vesicle which finally develops into a plastid. Proplastid, when exposed to darkness, will develop into etioplast. Etioplast is that type of a plastid which has not been exposed to light. On the other hand, proplastid, when exposed to light, will develop into chloroplast. Now, as we were discussing, that chloroplast is a semi-autonomous organelle. Now, what does the semi-autonomous imply? Semi-autonomous means what? It can perform majority of its vital functions by itself. It can work by itself. It has its own DNA, it had its own DNA, RNA and its protein synthetic machinery. The ribosome present in chloroplast is of 70S type. Now let's talk about the structural details of chloroplast. Chloroplasts of higher plants have disc-shaped or oval double membrane bound structure. The length is approximately 10 micrometer and the diameter is 2 to 4 micrometer. The algal chloroplasts are normally larger in size and of varied shape, like some organisms have spiral, some have cup shaped, others have circular bands of chloroplasts, like that. Sometimes polyploid plants also have chloroplasts which are larger in size. Several chloroplasts occur in each cells of higher plants. In algal cells it varies from 1 to 2 per cell. The cavity enclosed by the double membrane is filled with a gel-like stroma that contains flattened disc-shaped thylakoid. Each of these flattened structure is known as thylakoid, and this entire pile, this entire stack is known as granum, singular granum, plural grana. This is also known as cranum, and it looks like a stack or a pile of coins with a diameter of 0.5 to 2 micrometer. This is the most important aspect of this particular slide. Stroma is the site for light independent reaction, that is carbon fixation, and thylakoid is the site for light reaction. We will not talk about the functional details in this particular lecture. We are emphasizing on the structural details here. Now let's look at its diagram. I've drawn a very simple diagram over here to give you a brief idea about its structural details. This is your granum, the stroma, and thylakoid. Here comes the outer membrane, the inner membrane, and in between we have the intermembrane space. It has starch grains in it, ribosomes which are of 70S type, and it has its genetic material. That means it has its chloroplast DNA, it has its own RNA like that. That is why we call it a semi-autonomous organelle. Spherical particles of 8 to 11 nanometer diameter emerge from the surface of thylakoid membranes into the stroma space. They contain the CF1 protein complex that is known to play important role in ATP formation. CF1 is attached to the thylakoid membrane by a complex of hydrophobic proteins called CF0. The complex CF0 functions as a transmembrane channel through which protons pass to the CF1 ATP synthesis take place. Now, I would urge you to please study the transfer of electrons along with this particular lesson. We are not emphasizing on the functional details and all photosynthetic activity related to chloroplast or the electron transport related to the thylakoid membrane or ATP synthesis, because we are emphasizing on the structural details over here. But it will be definitely easier for you to understand if you kindly go through these few topics once before you study about this particular slide. It will really help you to understand better. See, this is one unstacked thylakoid. These are the stacked one. That means what? Granum. You have your PS1 and PS2 over here in the membrane. Then you have your CF1 and CF0, which takes part in ATP synthesis. This is your thylakoid membrane. This structural detail can be tallied with your text, which will give you the knowledge about ATP synthesis, electron transport. So it will give you a better picture. You will understand that better. So this is all about our structural details of chloroplast. I hope you have enjoyed the lesson. If you have liked it, please do hit the bell icon. Stay updated with my upcoming videos, and please utilize the time and make best of it. Thank you so much.